Hi, my name's Koki, and I'm the Chief Scientific and Technical Officer at Atara. And this is a 15-minute talk, but hopefully you're going to get to know our story through the three products I'm going to touch upon. 2-second summary is we're an off-the-shelf T-cell therapy company, and I'm excited to tell you about what we do. So I think just the top note for us, and this kind of hopefully came through the panel, we're in that first wave of companies in the T-cell space for off-the-shelf. In fact, we're the first to obtain approval for an allogeneic T-cell therapeutic, and this was in the EMA, and that was December 2022. And that since has been commercialized with PureFab, our partners in Europe. I'm also going to tell you about our MS program, so I'm going to go from ultra-orphan into a big indication, which is MS, and we have a lot of excitement about that. We have a primary analysis that's going to be in a few weeks. And then the last is our foray into CAR-T, because we still believe in the value of the off-the-shelf program. And everything that we've learned from tab-cell, we're going to apply it to CAR-Ts. So let me start here with the problem statement. I think the problem statement really is that we think cell therapy is the answer. And I think a lot of us grew up with small molecules and antibodies, T-cell engagers, but it still goes back to the effector immune cell, and we think that's the T-cell. So our platform is based on healthy donor T-cells, and what we do is we access that EBV memory T-cell in healthy donors, we expand them, and that's tab-cell. It's a similar process for 188, and then for the CAR-T, the nuance is that we transduce a chimeric antigen receptor. And I think there's been a lot of approaches. Ours is a little bit orthogonal, so what we do is we eschew gene editing, so we're taking a transplantation approach. So we're doing partial HLA matching, and it's a lot simpler for us from that standpoint. We're letting the T-cell receptor do the work, and the partial HLA matching figure out that very tough biology for us, and I'll walk you through that. This is what our pipeline looks like, and again, it's sort of risk-balanced. It's commercialized in Europe. It's phase 3 in the US, finally a BLA forthwith. This phase 2 program in progressive MS, and then we have a set of CAR-T assets. One of which IND was cleared by the agency. It's been a hot minute, but it was recently. It's like 2 months, sorry. Okay, so let me tell you about Avalo. And let me just tell you, this is a program we could not afford to develop. And again, it's a T-cell therapeutic for EBV-associated ultra-rare diseases. It has breakthrough status in the US, and it's been approved in Europe. Let me just tell you a little bit about the disease and the therapeutic. What we're faced with in these patients are transplant patients often face these post-transplant complications. One of them is lymphoma that arises because of EBV, which is in all of us as adults. The problem statement is there's a frontline therapeutic, but 50% of patients fail, and then often they succumb within 30 to 40 days. So the problem statement really is, what are you going to do? And in the history of the program, if you think about it, what physicians used to do was donor leukocyte infusion. And in DLI had graft versus host that's 40 to 60%. So at Memorial Sloan Kettering, what they devised was tab-cell, which is an EBV-directed T-cell therapeutic. So this has over 20 years of clinical experience, and really our paradigm is 3 days. So a patient presents, they failed frontline, and we can get the drug to patients in 3 days. The key nuance, which is different from what I think we're used to, is there's no lymphodepletion. So it's pretty simple that way. And there's no ICU or CRS or ICANS to worry about. So it's pretty simple as a therapeutic. And again, you could ask, well, if you're only serving 300, 500 patients in Europe, 300 patients to 500 patients in the US, why? And it's really about this slide. It's efficacy and safety. So the first thing I'll tell you is, again, the 2-year survival rate is greater. It's almost 90% if you're rounding like I do. I'm a biologist, so I'm going to round. The overall response rate on average is greater than 50%, and these are the data that we've shown ASH last year. But what's even more critical is when you look and you're able to get the drug into patients sooner, which is on the far right of this slide, which is expanded access in Europe, the response rate's obviously higher, right? Because you're getting to patients sooner with the same overall survival rate. So this is why we're seeing tab-cell to the finish line in Europe and the US. We think that it's an amazing drug. It's amazingly safe. There's never been any safety issues. And so we were really excited to get this finally approved in Europe, and hopefully we'll reach the same point with the agency in the US. Next, I'm going to tell you, I'm going to go from ultra-orphan into big. And I think this is something that you're seeing in the CAR T field. I've only been at the company for 3 years, but I'll tell you, we've been in autoimmunity as a company for 5 years, okay? And this is this hypothesis that EBV causes MS. And it's kind of borne out in the science in the past year or so, that there's a very direct link between EBV and it's necessary and sufficient as one of the first things that can lead to MS progression, and obviously there's other genetic factors. That came out in Science and Nature. The first was a population genetic study from Harvard, and when you think about smoking and cancer, it's like you're 12-fold more likely to get cancer. With EBV, they found the linkage to be 32-fold. So there's strong genetic linkage, in other words, that once we all seroconvert and have EBV, this is one of the first steps leading towards MS. It was an elegant study. The other study from Stanford really showed that you get molecular mimicry, which explains a lot of autoimmunity, but they showed it in a very direct way. And so that's what this therapeutic is about. It's targeting latent EBV and autoreactive B cells in late-stage diseases where we've shown our data in progressive MS. And this is a little bit different from relapse-remitting. So in this space, there are no therapeutics which really improve disease. One of the approved drugs, for example, Ocrevus, can delay progression for about 6 months, but that's about it. So there's huge unmet need in this space, because after you reach the stage of progressive MS, the patients are terminal. So this is a lifetime of disability that just gets worse. Why we're excited about this program is that in our phase 1 study, 20 of 24 patients showed durable, stable disease or improvement. So 1/3 of the patients showed improvement, and I'll tell you how. But even showing stability in this segment is amazing, because there's no therapeutic that currently does that. And I'll tell you, the skeptic in me could say that there is some subjectivity to EDSS, which is a clinical readout, but we also have quantifiable biomarkers using MRI. So what you see is some evidence for remyelination. What you also see in the cohort in the phase 1, the stable disease or improvers, was actually a reduction in brain atrophy. So this is why Atara launched the phase 2 study, which is called EMBOLD. It's double-blinded, placebo-controlled, and those data are going to read out in early November. So where we've just gone in our journey thus far is we've gone to ultra-orphan lymphoma, to this big indication, which could potentially be, instead of hundreds to 1,000 to 100,000. So this is one of the things that gets me really excited working at Atara, just this ability to take this platform into autoimmunity. On that theme, the other aspect of what we do as a company is we take the same platform, which has been battle-tested in over 500 patients now. So it's off the shelf, it's allo, there's no gene editing, and we haven't seen any host versus graft or graft versus host. We haven't seen CRS. So we thought a good way to further leverage this and all of our learnings in GMP and commercial GMP was to go into the CAR T space, with a nuance, of course, because it is crowded. But let me walk you through that. So again, there's a lot of different ways to attack this. And I think similar to the antibody space in the late 90s, there's just a lot of approaches, and we don't know what's going to work yet. What I like about this approach and what drew me to the company was we're maintaining the T-cell receptor. And what that does is that helps you during bioproduction. So it's a physiologic stimulus. So in other words, you're presenting the cognate antigen, it's of the right affinity. So whatever you're getting in the end has the full repository of central memory, effector cells, stem-like memory cells. So that's one advantage. The second advantage is, we think, based on tab-cel and 188, and again, the 500 patients, is it obviates the need for us to do gene editing, which is incredibly complex, and it's not in our wheelhouse. So again, from a production point of view, but also the biology of EBV T cells, we're maintaining that T cell receptor. And then the rest is sort of straightforward. We use 1XX, and I'll show you the data for that on the next slide. We think that that's a tremendous advantage, not only because it's, our construct is CD3, CD28. Most constructs are 41BB. But it's also maintaining the ability to sustain cytolytic killing in a serial fashion. And obviously, we have armoring that we don't necessarily talk about a lot, but we do have our toolbox. So this, in a nutshell, is the CAR-T pivot for us as a company. This slide is a little busy, so let me just walk you through, and let me shake hands with the elephant in the room. Everyone will say, well, why are you also moving into the CD19 space? So I wake up every day of my life, and people I work with ask me the same question. And I'll tell you that, yeah, it's a high bar, but it's also the space we know the best. We know PK/PD, we know toxicity the best in this disease indication. The point of this program is we think it's good enough to be competitive, if not one of the best in class off-the-shelf programs. So that's really the gauntlet that I'm throwing down, right? And there's a couple of reasons why that is. The first is the manufacturing process that we have has tunability. So you have the innate advantages of a memory T cell, the EBV T cell, but then the manufacturing process, which can tune the T cell, which we know is important. My engineers love fold expansion. My biologists don't. The clinicians tell you want to see the T cells not expand in culture, but in the patient. And we've seen that in the auto CAR-T field. So that's what we've engineered into this program, in addition to 1XX. Now, the relevance of 1XX plus the phenotype, that's the middle panel, and you could say that's pretty academic, because really no one knows what memory T cell markers are. I'm an immunologist, and we're not the smartest people. We tend to fall in love with canon. And I'll tell you, we can convince ourselves that central memory is important, and we can show you the markers and the enrichment for this. But where the rubber meets the road is this very well established model for NALM6. So this, to use my Boston language, is a wicked aggressive model. Okay, in this model, typically your mice are dead at 21 days. And you could do a lot of tricks with multi-dosing or add cytokines or add T cells that are 50 to 1. This is a pretty high bar model. So when I started to believe that we had a platform was when we began to run these studies. So what you see in red is an auto benchmark, which is Tecartus-like. It's one of the better autologous CAR-T paradigms. And what we were seeing again and again, using this as our benchmark, that we could reach the point where we could generate an off-the-shelf T cell from healthy donors, which consistently could perform at or better. And so this is the program that we're going into first-in-human studies shortly. So that's our story. I always talk really fast, but I hope that the takeaway is that what we've done at Atara is we've taken this academic program, tab-cel, and we've commercialized it, which was not trivial. And we've had multiple interactions with the agency, and we think we're on track to get it to approval. And again, I'm not going to say that that's easy, but it's been a productive conversation. So we think that we'll take this program, which was invented and developed in the US, approved in Europe, and it'll finally reach American patients, and we're excited about that. At the same time, we're excited about this transcendent ability now to take these therapeutics and move into autoimmunity. Because oncology really, it's a very competitive space, but we think we can reach even more patients, and I think a lot of people agree with us. It's more than MS, obviously. We could also pivot to lupus, which we're also considering for our CD19 program. But that's what we're excited about. And as a scientist, again, it's the CAR-T franchise. As we learn how to develop these CAR-Ts, we're learning every day from our manufacturing of our commercial GMP product and our phase 2 product. And so we hope that next year will be very exciting for us. And thank you for your time.